we shall be discussing the working of an enzyme. As we know, enzymes are biocatalysts, well, little factories inside living cells, if you will. The factory on the screen is converting carbon dioxide to carbon monoxide. It uses some electricity, electron-proton gradients coming in from the wires there, to help make this conversion. Here, a hydrogenase is shown to be the source. The carbon monoxide produced is then sent to another factory, acetyl coenzyme synthase or ACS, for further processing. Unlike factories we see everywhere, this one can perform the opposite task too. That is, convert carbon monoxide to carbon dioxide with the same machinery. When that happens, this factory now produces an electron-proton gradient instead of using it up. This, as you'd have guessed, is a model, admittedly an oversimplified one, of the enzyme that we're going to discuss today. Carbon monoxide dehydrogenase.
But wait, this is not it. In fact, on the screen is a dimer of the CODH ACS complex, the cyan and pink ones being the CODH units and the blue and orange ones the ACS units. As we know, these units are bound together by non-covalent interactions between residues from the participant peptide chains, such as the ones shown here. Unlike the Calvin cycle, where carbon dioxide fixation is used as a carbon source and an energy source, CODH performs only one of these functions at a time. In microbes where carbon dioxide is reduced, CODH often coexists with the ACS unit so that the carbon monoxide obtained can be used to acetylate coenzyme A. Acetyl-CoA can then be used in further steps for carbon assimilation. Here, we shall be focusing on a single CODH unit. So let us get rid of the ACS units. CODH is usually found as a homodimer in organisms where this pathway is only used for carbon monoxide oxidation as an energy source. So we will be looking at only one of these units now.
In this nickel-iron variant of CODH, we can see three major catalytic clusters. These are called the B, C and D clusters. Each CODH unit has its own B and C clusters, but a single D cluster is shared by the dimer. In case you were wondering, the A cluster exists in the ACS part of the complex, which we shall not be discussing here today. The conversion of carbon monoxide to carbon dioxide or vice versa occurs at the C cluster. This cluster is deeply buried inside the enzyme such that the C, B and D clusters align as an efficient redox wire with 10 to 11 Armstrong intercluster distances. This allows for rapid electron transfer between the clusters. Another extremely interesting feature of this enzyme is the existence of a tunnel with histidine residues from the C cluster to the enzyme surface to allow the transfer of protons in and out of the enzyme. From the C cluster in CODH to the A cluster in ACS is a hydrophobic tunnel 70 Armstrongs long to avoid the loss of carbon monoxide during the transport.
We could go on with the interesting features of this enzyme, but we'll skip that and look into the C cluster where all the magic happens. We will be describing the carbon dioxide reduction, but each step being reversible means that we are effectively also describing the carbon monoxide oxidation. This cluster is an iron-sulfur distorted cubane with a nickel atom. An iron atom in the C cluster is outside the cubane and is known as the pendant iron atom. The pendant iron atom is coordinated by histidine, cysteine and a mu3 sulfido ligand and supposedly a small ligand, possibly even a hydroxide ligand, but this does not appear in the diffraction pattern, so it suggests a possible hydride ligand. This ligand needs to position itself so that nickel can attain square planar geometry without posing steric problems.
We shall now discuss a proposed mechanism of the enzyme function. For reduction of carbon dioxide to carbon monoxide, the CODH enzyme is in a single electron reduced state, where nickel is in a T-shaped geometry. The incoming carbon dioxide binds to nickel by a 2-electron reduction of the enzyme. Carbon dioxide acts like a triatomic bridging ligand between nickel and iron. These oxygens form hydrogen bonds with a histidine residue and a lysine residue respectively, stabilizing the complex. Carbon dioxide is activated as its LUMO attains back bonding from nickel and has to fit perfectly into the active site by bending the OCO axis. So, now the oxygen atoms have a higher partial negative charge which is stabilized in the complex by transition metal bonding as well as hydrogen bonding.
Consequently, cleavage of a CO bond results in the nickel carbon monoxide species as this oxygen abstracts a proton from a protonated histidine residue and repositions itself on the pendant iron atom. The nickel CO species is formed which dissociates to give out the carbon monoxide and the original nickel 2 center, which is ready for a new cycle. Coupling carbon dioxide reduction with water formation from protons brings down the reduction potential from a massive 1.9 volts to a manageable 0.5 volts, which is the reason why we showed electricity in our little factory at the start.
While the mechanism seems to be fairly simple, The genius of nature is seen all over this enzyme. From selecting and positioning both first and second sphere ligands, which by the way are conserved across variants, to designing those tunnels to facilitate the reaction and prevent loss of precious product. There definitely is a lot of chemistry to be learned from this enzyme.